in stem cell biology at Cancer Research UK and postdoctoral research training on autism at University College London. His current studies are at the forefront of a rapidly evolving field of investigation into the role that immune response plays in neurodevelopmental conditions such as autism. He was the first to demonstrate links between immune dysfunction and the severity of impairments that are hallmark features of autism, such as social interactions and communication. Overall, Dr. Ashwood has worked across traditional disciplinary boundaries to examine connections between different biological systems during development in order to understand how they lead to the characteristic features of autism. Specifically, he has highlighted the importance of innate immune pathways, gut immune brain connections, and the presence of autoimmunity in some children with autism. He's the author of over 100 articles on autism and has received recognition for his innovative work. Dr. Ashwood, please take it away.
Thank you very much, Pramila. Let me just share the screen. Okay, thank you. And, where am I? There we go. Oh, jumped ahead. All right. Well, thank you very much. Thank you, Pramila, for your introduction. I'll make sure I cut back the bio next time because that was too much. Thank you. It's an honor to be able to speak at the Brain Foundation. And give a little update on some of the information that we're doing. So today I'm going to talk about immune regulation and its issues in gastrointestinal symptoms in children with autism. Okay, there we go. And so we've heard from the excellent speakers this morning about gene and gene environment interactions, and I think from Sarkis and Joe about and Matt and Stuart about the fact that there's a very strong genetic link which links with high risk genes, and then a combination of maybe lower risk genes that all contribute to potential risk for autism spectrum disorders. And the same can be true for the environment, that there's potentially very high risk environmental agents that at this point we don't really know. Maybe valproic acid is one that springs to mind, but a number of others that may be, common environmental exposures such as air pollution, changes in the microbiome, or like with the genes, you can get combinations of the two. So you can get infection and you can get maternal infection, for example, in some of the animal models that Sarkis mentioned earlier, and the microbiome. So you can get combinations of the environment and combinations of the genetics. And somewhere in between, maybe this is what is the most likely, or at least in my opinion, the most likely causes of autism spectrum disorders. And at that interface, you have maybe the immune system. It's uniquely situated where it deals a lot with environmental exposures, and is, of course, most of its function is predicted by genes, and changes in gene polymorphisms or epigenetic dampening down of gene signals can change the immune function. So this is where I've always been interested, but I always want to make sure that we start this any talk with the fact that the immune system is incredibly important, and it's important to mediate resistance to infection or to eliminate infection. In fact, I guess we all know that now because we're living in the age of COVID and the importance of protection against COVID, and the development of things like herd immunity, so that the more vulnerable in society are sort of protected against that. But we have this complicated and complex system where a lot of cells from the immune response interact to eliminate and protect us from things that are potentially harmful and potentially fatal. So it's always important to think about it in that way. But when things go wrong, you can get immune responses, and again, that become apparent, you become problematic. So again, if you think about COVID, this idea that there could be a cytokine storm, the release of mediators that cause further damage or the most damage to an individual, and could lead to progression of disease, of the infection. You also to the fact that some of these immune mediators could lead to issues such as the long-term COVID, where you get fatigue and all sorts of extra chronic symptoms that can last for a very long time, and we just don't know how long that is. But when thinking of autism, we can see when things go wrong, people have shown evidence that there is inflammation in the brain. Andy Zimmerman and Carlos Pardo produced some of the first work on that, very important work on that, about showing that there were increase in cytokines in the brain in individuals that had autism. More work, recent work has shown that there's cells of the immune system in the brain, the microglia, that they have become activated and that they can produce factors that could attribute to changes in neurological function. We have a speaker, Dr. Fasano, later has also shown that there's altered blood-brain barrier function in the brain, and we had a speaker last year who's talking about the fact that they could be T cells that come from the periphery that get into the brain, and that that could then lead to a progression of sort of inflammatory responses. We also know there's a lot of common comorbidities, immune comorbidities in autism, such as allergies and asthma, and in particular interest to me is things like GI dysfunction. And again, I don't know if Dr. Kriesman will talk about it more, but the increase of inflammatory bowel disease has now been set as the population of children with autism age. The prevalence of inflammatory bowel disease in the autism population has been shown to be increased. When thinking of immune cells and immune function, we've seen that there's differences in antibody levels, there's differences in monocyte cells, some of these other immune cells that are very important for dealing with different functions of infections or involved in asthma and allergy and other things. They've all been shown to be dysregulated. And I think many of you probably know that there's this increase in antibodies directed towards proteins of self, so your own protein structures. So what I'm interested in is how potentially the balance in the immune system, if it shifts one way or another, how does this affect the nervous system? And we've shown that sometimes increased inflammatory cytokines, this has been linked with worse behavior. So there's increased, so as these inflammatory cytokines increase, social behavior gets worse, communication gets worse, and we often see more increased restricted repetitive behavior. So, but that's not always to say that increases is the only way. You can also get the fact that the immune system can decrease, and this could also have effects on the nervous system. So some immunodeficiencies could also have a serious effect. So you just got to get it right at that tipping point, and otherwise the two systems can interact and change. And also the nervous system can have a big impact on the immune system through production of neurotransmitters and also and signals that can tell the immune system whether to activate or not. And this is maybe how things in the microbiome can be producing neurotransmitters that affect both the nervous system and the immune system as well at the same time. So as an immunologist, why is it important for me to sort of think about mucosal immunology in particular? And this is how I first got into the field. And it's the most immune cells in our body are within the GI tract. So this is something that is you have more immune systems in follicles. You have more immune cells within the lamina propria, and this outweighs cells in the rest of the body. And what to me is very unique is that there's a separation of what's going on in here in the lumen of the gut, which is bits of dietary factors, bacteria, and all of these immune cells that are present underneath that one single cell layer of cells. So it's not a big barrier. It's just there are some protective reasons why this barrier is effective, but it's not very. The difference between host and environment is very small. So this to me has always been a fascinating thing to look at, and also to me is why don't we react to everything that we take into our body, because it is essentially a foreign element that comes into our body, and there are checks and balances in place to protect us from reacting to all of these things. So that's one of the ways that I look at these things, and how I've always been interested in it. And then when I moved from looking at Crohn's disease into looking at GI issues in autism, at that time there were a number of early studies, and the first study is really to go back and look at the original description of autism. And if you look at that, you'll see that at least 8 of the original number of subjects that were described, at least 8 of them had issues that could be described as GI issues, such as vomiting, eating issues, food issues, selectivity, and so it was always there. And then further over the years, in the 70s, people started looking at things like glycosylation and showing that there's differences in sulfation. So some of these things could be related to the microbiome, or they could be related to the host themselves. Bacterial overgrowth, diversity or lack of diversity of microbes are certainly things that we've seen more and more frequently. We haven't really got a consensus of which bacteria are different, or which groups of bacteria are different, and that could depend on dietary factors within different populations as they're measured, or could also relate to picky eating in certain subjects, and selective eating or food sensitivities in certain groups. But we've also seen that positive interventions, dietary interventions, could lead to positive outcomes in certain patients. So the estimates of GI issues vary quite dramatically from whether you're looking at a population-based study or whether you're looking at a clinic-based study. But it's very clear that a good proportion of children with autism have some sort of GI issue. And I think when I first started this, 20 years ago, people didn't believe that, and now I think it's pretty well established that a good proportion, a third to a half of people, depending on which report you look at, have GI issues. And the main complaints are things like abdominal pain and constipation and diarrhea. And I'm sure Dr. Kriegsmann is going to talk a lot more about this, and especially these sort of findings here, when this is the ileum. And this is a look from a colonoscopy, that they've seen that you get these formations of what's called lymph node hyperplasia. So these are lymph tissues that have expanded, and within that, a lot of T cells and B cells that are expanding and overreacting and over-responding and proliferating. Also, this has been seen in the colon. You see these quite distinct, these sort of responses. And then if you're looking at H&E staining, you'll see there's a lot of activity going on there with a lot of immune cells. So some of the really early work that we did God, that's nearly 20 years ago, that we looked at. It was taking sections of the gut from individuals that had autism and typical developing controls, and seeing how many immune cells were in that tissue. So if you're looking at a typical developing child, you'll see that there's some. These are protective, and they're there to protect the host from what's going on in the lumen. But in the child with autism, there's a lot more of these immune cells stained in brown here. And that there's, when you look at the levels of these cells, that they're increased above the controls and similar to established inflammatory disease. They're not the same, but they definitely an increase in these cells. So when we looked at individual, these markers just denote T cells that are present, and we found that in different compartments of the gut, lamina propria, which is that level below the epithelial layer, and in the epithelial layer, that there's an increase in the number of immune cells within the gut in autism over typically developing controls, and same as inflammatory celiac disease. And this we found in a number of, we found it all the way through the gut. So we found it in the esophagus, we found it in the stomach, we found it in the transverse colon, and then the intestine. So it seems that it's not distinct in certain regions, which other things like celiac disease and inflammatory bowel disease are sort of contained within different regions within the gut. But this was a pan-enteric sort of inflammation, which was subtle, but it was definitely an increase in those immune cells. So the next phase of, well, we've got all of these immune cells and we've got an increase in these immune cells, but are they actually doing anything? Are they producing anything that could be harmful or could lead to this inflammation that potentially we're seeing? And here, again, in some of the early work, we looked at whether they were producing different types of inflammatory cells. And, you know, the immunologists like to divide things and like to group things into different categories that not always fit easily, but you know, to help you differentiate what might be a certain process in the immune response. So here we looked at things that were Th1 cells, and this is thought to be involved in inflammation and cell-mediated responses, and also for Th2 and regulatory T cells that are involved in potentially, if they're increased in Th2 cells, they're involved in sort of allergy, asthma type responses. So what we found is that in children with autism, so in the yellow, we had an increase in these pro-inflammatory cytokines. And again, because of things like COVID, we got more familiar with some of these terms, but like IL-6, this is an inflammatory cytokine, and TNF-alpha, another inflammatory cytokine. And these were increased, and they were increased not to the levels of Crohn's disease, and that's very important, but they were increased above non-inflamed control. So this is actually terminal ileum. So the children who are coming in, who are the controlled children, they're coming in with complaints of GI issues, and potentially they have inflammation. But these were controls with that had things like constipation and functional GI disease, which seemed to be an appropriate control at the time. But we also saw an increase in these Th1 cytokines, IL-12 and interferon gamma. What was interesting is that when we looked at the cytokines that are involved in allergy and asthma, we saw that there was an increase in these, which wasn't there in the Crohn's disease, such as IL-4 and IL-5. But when it comes to the regulatory cytokines, such as IL-10, we got this massive decrease compared to both inflammatory bowel disease and control. So what I took away from that, and what we thought, was that there's an increase in inflammatory, but a decrease in this regulatory signals. So what we also found is that what this may mean is that we've got this increase in inflammation, that it could have an effect on the barrier function. So we looked at the barrier, and then one of the ways we could look at is whether there was an autoimmune reaction at that barrier. So we have antibodies, IgG, that bind to we found that bound to the inflammatory, they're bound to the basement membrane of the epithelium. C1q, you just heard a little bit about complement proteins. When these two combine, that they form an attack complex that can lead to tissue damage. And what we found is that when we co-localize this on a biopsy from a child with autism, that those two coexist and they co-localize on that barrier, potentially meaning that this could affect the epithelial barrier function. So we're very interested in how this this could develop, and you know, this was work that I did as a postdoc, and I wanted to carry it on. And here we're looking at, and the hypothesis was to do with this idea that there is increased inflammation, but there's decreased regulation. So we wanted to look at the tipping point between these two things, of what, whether in children with GI issues, whether there was this increase of these certain cytokines and a decrease in the regulatory certain cytokines. So it's that regulation that I'm really keen on. So we looked at different groups, autism with GI issues, autism without GI issues, typically developing children with without GI issues, and typically children with GI issues. And this is what we did with Sarkis and Alicio, who's going to talk later, and Paul Patterson. And so we started looking at what we'd take the blood, we'll isolate the plasma from the blood, we'll isolate immune cells, and we can stimulate those cells to see what they do. I mean, it's interesting to see a cell at rest, but it's also interesting to challenge it and see if it has a different type of response. So we could stimulate cells, see what cytokines they're producing, and also start looking at flow cytometry. And flow cytometry allows you to look at, does that individual cell change? Does it have certain things on the surface that shows it's activated or quiescent? So what we found, which was interesting, is that when we started looking at groups, when it came to pro-inflammatory cytokines, that we did see not only differences between children that were typically developing with no GI issues, but we started seeing potential differences between kids without GI issues and kids that had GI issues, that there were differences in pro-inflammatory profiles, but generally autism was more different than the typically developing children, and had more activity to their once they were stimulated and then produced. Potentially more interestingly, when we looked in terms of the GI issue, when we looked at regulation, we found that there was a big decrease in the amount of this cytokine that's very important in immune regulation, transforming growth factor beta, that it was decreased in the ASD GI group compared to the ASD no GI and the typical controls. And that's at rest. But when you also stimulate them with different activators, so activates T cells, this LPS is important in responses to the microbiome and bacteria. And you found that there was this big decrease in these ASD in the ASD group. So we also wanted to keep going forward and looking at different types of cells, like we mentioned earlier. I brought in the idea of different Th1, Th2, and regulatory T cells. And what we found is that there was an increase in the group with ASD-GI, there was increase in these Th17 cells. Now, Sarkis alluded to the maternal immune activation model and how in that immune cells are important, maternal immune cells are important, and also cells in the offspring that could lead to GI issues. And what's interesting is that the paper, the most recent immunity paper, has shown that there's this increase in this animal model as well, which is very interesting, of Th17 cells. And we found the same here in humans, that there's this increase in Th17 cells, and also Th17.1 cells, which are those that produce IL-17 and also produce interferon gamma. So we're getting some parallels between an animal model and humans. So we also then looked at whether there's a decrease in these regulatory cell subsets by flow cytometry. And here we saw, again, that there's this big decrease in the ASD-GI group compared to typical developing controls. What was also interesting is that we did see differences in behavior, and I'm sure that Arthur, Dr. Kriesman is going to talk about that as well, that in the ASD-GI group, there's more, it seems that there's worse, more impaired behaviors. And we saw that these were linked to immune responses. So to me, what's really interesting is this key link between immune activation and, more importantly, immune regulation. And I think that's where things sit, that there is a decrease in this. It's important to maybe up the regulation to be, because at the moment it's way too low. So one of the projects that the Brain Foundation is supporting, and thank you for that, is that we're going to look at this epithelial barrier. And as I say, to me, it's amazing because it's just this one cell thick barrier, but it's very important how it, and there's various ways of how it can educate this regulatory immune cell population. So we want to look at this epithelial cells and we can take them from individual patients and we can grow them out in epithelial organoids, much like we heard discussed about cerebral organoids. We can take the immune tissue out and we can look at the inflammatory responses, we can look at cellular stress responses, and you know, again, look at what would happen when we challenge them with certain things like bacteria, what happens when we challenge them with different metabolites, what happens when we challenge them with different microbes. And so here we can have this, we can take the immune cells and grow them. And it always fascinates me that you can get these amazing pictures that look a little bit like the villi that you've been produced. And we're focusing on the ileum, terminal ileum, because that's where we think a lot of the immune responses are occurring. So we've taken a lot, with the support of Brain Foundation, we've started biobanking certain pediatric patients from autism and from controls. And we've also taken some different cell lines from immune activation models and also some other models that might be important in autism. And the CHD8 model is important with GI issues and with immune issues in autism. We're going to run some single cell RNA-seq because there's going to be different types of immune cells within those populations of the organoids. And like I say, we're going to see what happens when we co-culture with different, from the same individual patients, co-culture with immune cells, with immune products, also with bacteria, if we can find a way of growing up bacteria without losing too many of the species within, when we sort of grow them out from patients, and see what the responses are of those epithelial cells. So I think this is very exciting, and hopefully that's where we're going to, this is the direction we're moving in. And I'd like to thank people in my lab. I'd also like to thank my collaborators, and I apologize for not putting on your names, but for Sarkis and Alicio, and we've got projects that hopefully we can get off the ground with Dr. Kriegsman and Dr. Walker. And of course, I'd like to thank the Brain Foundation for their support. So thank you very much. And can I stop sharing? Stop sharing. Okay, great.
There is one question on the chat. I think it asks, the anti-TNF drugs are used to treat Crohn's GI. Do they work in ASD?
I think that's probably a question for Dr. Kriegsman later, and it would be, you know, it's one of the things that I mean, one of the offshoots, I think, of the COVID crisis is that there are a lot of antibodies or antibodies out to different types of cytokines. So there's definitely a lot of now that there's these reagents that could look at how to change the immune response and immunosuppression based on specific cytokines. So yes, I mean, whether TNF is the right target is something that we have to look into. But I think, in my mind, I think we need to increase the immune regulation, and there's ways of doing that in terms of that you could potentially take sort of cord blood, for example, if that's stored, and grow regulatory T cells from an individual to be able to then give back. Let's see what else. There's a question. Is there a test that you can quantify base inflammation in a child? So again, there's not necessarily a, people have looked at that. I mean, what we haven't been able to look at is whether there's levels of cytokines, and whether there's a way of looking at population normals, if you know, normative ranges. And so things happen in ranges, things happen at different ages. So different age, you're going to get a different immune response as you go through. So we need to have, if we're going to do that, we need to have larger subject numbers to be able to have something that's informative enough. And I worry about, you know, it's the same with any of us having a blood test, is that it's a snapshot in that time, and that you need to probably look at a longitudinal effect of what your T cells were, what your cytokine levels were. So we're kind of a long way away from that. But yeah, that's an interesting question. Okay.
I think Dr. Kriegsman raised his hand.
Sorry, once you start looking at, there's the Q&A, and I haven't got to the chats yet. Arthur, did you want to say something?
Yeah. Okay. Can you hear me?
Yes.
Yeah. Okay. Great to see you, Paul. Okay. First of all, thanks for the lecture. But just a basic question that I have is that, why is there no commercially available test that clinicians can use in advance of using a biologic agent like a TNF-alpha antagonist? Why is there no biologic test that we can do to identify those patients that do have elevated TNF-alpha versus those that have a more of a predominant IL-12, IL-23 type of profile, since there are drugs that are currently available that would be more effective in one versus the other? Why is that, at least to my knowledge, still relegated to a laboratory investigation and not commercially available?
Well, I think it comes down to the fact that there's nothing in the blood necessarily that in plasma or sera that's an easy target to look at. I mean, most of what we found that gives you the best differentiation between controls and individuals with autism is that you need to do something with the cells. You need to stimulate the cells and then be able to look at the the stimulated responses. And I think what's interesting is that even within that, you have, and I hinted on it, and I did, maybe next time I'll show some more pictures, that with kids with GI issues with autism, they have this profile where they have increased IL-17, whereas kids without GI issues and autism, they have an increased IL-4, IL-5 profile. So they have, so you have a Th17 response and a Th2 response, but they're both up compared to controls. So, you know, we just, again, it's one of those things that I think we just need bigger numbers. And, you know, it's the same with how anybody established any sort of baseline levels of just what's high CD4, what's high, you know, T cells, because they were able to measure quite a large number of a group before. And once we've got that, then maybe, you know, then maybe you can move on to the next thing. Or, you know, hopefully we can do things like we can create certain things in a dish from iPSCs, let's say, and then you have a mini gut that you can be able to say, here's a response. And, you know, that's something that then the clinician, it will inform the clinicians a bit more.
Interesting. One thing that Dr. Walker and I are working on, and I don't know if he's going to mention this today, is we do have an organoid biobank of enteric tissue. We've recently started that. You may be aware of that, actually. And the other interesting and clinically relevant issue is that I would imagine that if there were a repository of, say, 1,000 ileum specimens from 1,000 ASD patients with GI inflammation, that perhaps I'd imagine would be, and versus controls, that would be a great starting point to identify which pro-inflammatory cytokines would be best targeted, either by existing pharmaceuticals or ones yet to be invented.
Yeah, I think that's definitely something that we need to you know, further, and hopefully the things that we might work on together will help us get some answers to that.
Looking forward to it.
Okay, so
we can move on.
Pardon?
We can move on to the next.
Yeah. Okay. What I'll try and do is answer the questions offline, or I can type answers into certain things in as replies. So I have no idea who the next speaker is. I've never seen them before in my life. Obviously, sorry, poor joke over Zoom. But yeah, I've known Dr. Kriegsmann for a number of years. He's one of those pioneers, if you like, that have, and he, you know, there's been a lot of obstacles that he's overcome, but he has persevered, and he's fantastic and brilliant, and one of the few pediatric gastroenterologists who are willing to see children with autism and to investigate their GI inflammation. Not many people do, not many pediatric gastroenterologists do, but he definitely, Dr. Kriegsmann has been a major pioneer in this area, and has, you know, as I say, there's been a lot of obstacles and issues that he's had to come through, but he has not only done that, but he's presented to the US Congress about his findings, and presented at major meetings, and presented to influencers, and has hopefully, you know, he will be now, you know, some of the work that he's been doing over the few years will finally, people will take on board, and you know, come along with him on some of the things that he's been trying to pioneer.